First, I'd like to introduce the panelists. So Gemma Hopewell is a professor at the University of Oxford. She's been really involved in a lot of aspects of cardiovascular research, thinking about imaging, machine learning, and also putting together the UK Biobank data set, which I think, as most of you know, is really the sort of most comprehensive biomedical data set in existence right now. Also with us, we have Brett Kroenke. Brett is an assistant professor at Vanderbilt University studying the penetrance of rare variants in cardiovascular diseases. And we have Junaid Nashad, who is a computer science grad student at the University of California, Irvine, working particularly on machine learning in image and genomics processing. So yeah, I'm really glad to have all of them with me today, and I'm really appreciative that they took this time out of their schedule. Again, for the audience, it's good to see all of you. I'm glad you were able to make it. And if you have questions, please pop them up in the chat, in the Q&A, wherever you can. For those of you who have my cell phone number, send me a text, whatever, and I'll try to ask the questions. I'll try to ask questions as we go along or in our Q&A session at the end. This probably will go for about 45 minutes, and we'll try to range topics all the way from really sort of general cardiovascular stuff all the way down to specific technical applications and all that. So hopefully everybody gets lots out of it. Like I mentioned, we had one on neurodegeneration last month, and people were very excited. So hopefully you are too. So with that, maybe I could just kick off some questions. So Gemma, why do you work on cardiovascular disease?
Wow, start with something easy, shall we, Ben? So
yeah,
great to be here with you. Thanks for the invitation. I mean, one of the biggest killers out there. I'm a population health scientist that works on precision medicine and large-scale epidemiology. And so to me, these are some really big and really important questions that affect a lot of people's lives. And I think to, in some small way, contribute towards making those lives better in understanding the causes and the consequences and the treatments for cardiovascular disease kind of gives me a lot of motivation. I think we're in a great time, we're in a really great era where we have genomics, we have multi-omics. So it's all that fast moving area becomes a really motivating and insightful area to work in. So yeah, lots of important questions to answer, lots of people that it affects, and some great people to work with collaboratively as well.
Awesome. So you mentioned precision medicine. What does that mean to you in the context of cardiovascular disease?
So I think it's about making sure that people get the treatments and in the best way possible. So the people that are going to benefit most are going to make sure that they get them, and that we don't necessarily disadvantage people by not giving people medicines that could potentially just not have been tested in them. So we can kind of help understand from using genetics and other omics data who we might be best placed to do things, and how we can hypothesize and how we can design clinical trials better. And so I think precision medicine is about kind of making the world better for everybody, and making sure that we think about individuals as well as populations. But I think generally we end up with a lot of treatments that are good at population health levels, and we should be making sure that people get these treatments, but we might be able to tweak some scenarios that make them better for particular people's circumstances. And you see that a lot in cancer, which isn't an area that I'm an expert in, but you see that kind of scenario a lot there.
So Brett, actually, following up on that like trail of using the wrong drug. I mean, you work on penetrance of a number of variants. So can you talk a little bit about how using drugs for inappropriate people, how particular, rare, and even common variants sort of affects that?
Well, I mean, we're in a, as Gemma was alluding to, we're in sort of a growth phase in the amount of genetic data that we're acquiring. You can imagine some of these Mendelian monogenic diseases, were discovered in individuals under extreme ascertainment bias. And you might find a variant, and you hear about this all the time. You find a variant that was 20 years ago thought to be this, really highly penetrant thing, and it turns out it's somewhat more common in the population. You just had an extreme bias going, which is why you observed it enriched in say some of the cases. And so, I think of it as like, what using all the information we have available to us, how accurately can we predict the risk of a phenotype, and specifically a phenotype people don't want? And then having established as accurately and as precisely as possible what that risk is, what's the appropriate pathway to mitigate that if that number is too onerous for the individual?
So early in that response, you mentioned sort of a preponderance of genomic data that's available right now. So I'm curious, why are large data sets, maybe large public data sets, important to you?
Well, I don't know. It feels like there are lots of ways of going at that. I mean, you can't, sort of a interesting, genetic structure is that there's a huge accumulation of sort of one-offs, or just a few people carrying certain variants. And so in some extent, you're sort of up against that, even sampling in very large populations. But, as you sample more widely, you can get a better sense for certain variants as to, what's the probability that it truly associates with a particular phenotype. And for me, my work is I'm really interested in the rare variant space, but I can't really do that ab initio. Like I need to calibrate it based on something. And so larger data sets that have becoming a greater sampling of specific variants that have, say, features like functionality or structural features or evolutionary conservation that can then be mapped onto that probability landscape, allows us to sort of reach into that more rare variant space, and sort of use that calibration of our understanding of these variants that are seen more frequently into how that might be translated into the rare variant space. And so for me, it's really critical to have several variants, so you can devise a robust method to predict all variants. And I think these data sets that are coming available now are also really helpful because they're, I like to think that there's no such thing as an unselected population. It's all biased in some way. But these are less so. I mean, I work up in the 12th floor here, a bunch of cardiologists in an arrhythmia center, and the people that are seen here, you might be drawing from a population that's as wide as the middle Tennessee or tri-state area, and so your effective sampling is extremely skewed, and so the numbers that you come up with are somewhat unreliable to first order, and so to have a ballast that you can use, which is a more a population that's more thoroughly being sampled rather than just ascertained and selected for a particular phenotype, helps us sort of sanity check the measurements that we're coming up with, and it also sort of add to this iterative effect or iterative method of trying to build those observations into how we then understand rare variants, because that's really kind of, as you mentioned before, that's sort of where I think my major focus is.
So to kick it back over to Gemma, you've in this space, you've also really integrated lots of imaging and then thinking about other types of features. How do you use like that other data to really paint a more accurate picture of the clinical landscape?
So first of all, I'd say I'm not, I think the value of imaging is fantastic. I'm not an imaging expert, but I think what I have put a great amount of importance in is phenotyping. And I think that imaging, the electronic healthcare records, so the likes of UK Biobank, well, I say the likes of it, there is nothing else like UK Biobank. It's, as people say, it's a gift to the world. But phenotyping is hugely important. And I think for understanding the biology, from thinking about kind of the maps to mechanisms, scenario and the pathways to patient care, you have to get that phenotyping right in the first place, because that's the thing that kind of then helps you think about the next steps. And kind of that's again that kind of precision medicine and that balance between phenotype, genotype, putting everything together. It's all very synergistic. So not just imaging, but all types of big data and small data for that matter can be important.
Okay, that's awesome. So I mean, I think one could think that sort of the extremely detailed level of phenotyping in something like UK Biobank is even a bit intimidating to folks. But I mean, you seem to think that's pretty valuable, and obviously it's a little different than many other public data sets. Can you talk about a couple examples of really sort of specific phenotype and how that can be used for cardiovascular research?
Sure. I mean, I think it's the value again in kind of bringing the different data sources together, and then kind of understanding greater depth as you move forward. So something like many people will have this in their data sets to have self-reported data. Now that can have many problems with it, because for different types of disease outcomes, how well validated that would be quite different. Luckily, people tend to know if they've had a heart attack, for example, or they've got potentially a rare disease, maybe thinking about Brett's area or other people's areas. People tend to know maybe if they have atrial fibrillation, another area that we work in, but also then synergizing that with information from things like ECGs. So as we have in UK Biobank, we have resting ECGs, we have exercise ECGs at baseline, and then actually supplementing that with primary care data, or actually something that's kind of a bit more chronic, like atrial fibrillation. It doesn't tend to get diagnosed in hospital. It tends to be something that you probably see in primary care first. And so being able to kind of, again, synergize those different data sources, bring information in from ECG records, because some people just don't know they have a particular type of disease. That's a really good example of where bringing the things together, the different aspects, is important for really capturing all that information. I think hopefully one area that maybe we'll talk about later, but kind of looking at patients' trajectories over time as well, and kind of building that in, understanding recurrent events, seeing how different diseases impact on patients' journeys over time, and multimorbidity. Sorry, lots of concepts there, but
that's awesome. So I do want to come back to the longitudinal analysis piece, and it's so critical and such a gift from UK Biobank and some other notable data sets. But what I wanted to talk about a little bit first is you mentioned, primary care notes and all kinds of other things, ECGs, for example, and clearly that gives you sort of a wealth of features that may not have even been expected, right? And so can you talk a little bit about ways your group and others are sort of deriving features from some of this kind of primary data, if you will?
Sure. So I think there's a lot of hard work, and I think it really reflects, again, a really great team environment, and bringing together multidisciplinary teams is really important. So we get to work with people that are focusing very clinically in their particular area. So electrophysiological disease doctors that can bring their input into it. So literally going through material, trying to understand what procedures, what drugs maybe are specific to a disease area, again, bring in additional amounts of synergistic information. So there's that angle of things, so the more human angle, if you will. And then there's a case of automating. I'm a big fan of automation, and kind of where you can develop algorithms that they're supplemented with human input rather than it being one or the other. And so a lot of people have been looking at ways of kind of getting more phenotype information out of things like the imaging data, developing training sets, and then kind of developing machine learning algorithms. I mean, Stephen Petterson's group in London, and some of the other panelists will probably speak better to this than I will, but kind of enhancing what's already there. I think that's the great thing with data, isn't it? You can gather the data together and then you can put together some kind of cool techniques and bring the science and the methodology on, and really increase that those training data sets, and think of new ideas and hack away, so to speak.
So Gemma mentioned machine learning. Brett or Junaid, do you want to speak to some of the ways that you can apply machine learning to some of these data sets to extract features?
Yeah, so absolutely. Whenever you have these like very large, diverse data sets, one of the, I think, first ideas that comes to mind is like, how can we apply machine learning to this data? And so one of the things that we focused on at DNA Nexus is not just trying to build classifiers that perform very well, but also focusing on interpretable machine learning such that we can then do further downstream analysis. So in the case of working with ECG data, one of the things that we wanted to do was we wanted to take all of the participants within that had ECG data, and we wanted to subgroup these participants and then perform GWAS in order to identify like common genetic traits in these subgroups. And so the way we tried to approach this end goal was through the use of interpretable machine learning, where the model is able to identify the most important features for each person. And then based on the overlapping important features, we can subgroup these participants and then perform GWAS. So there's a lot of different directions that you can take in terms of like what kind of like high level information you can extract, and then what kind of like further interpretation you can provide to clinicians. So it's more than just like finding classifiers that get really good accuracy, but you can actually do a lot of like further downstream analysis as well.
Cool. Can you talk a little bit? I mean, I think that's you touched on something that's really powerful, which is thinking about how to take machine learning results and communicate them to clinicians. So can you talk a little bit about your thoughts on taking machine learning results of machine learning algorithms and communicating them to the clinical community?
Yeah. So as in terms of like communicating results, why I think one of the best ways is through like visual analysis. So if you're working with, say, imaging data, if you can, if you're trying to develop interpretable models for imaging, then something you can try is like using things like class activation maps, so that you can easily display like heat maps, so that the clinician can easily look at like a case where the model makes a false prediction, and then they can at least identify like, okay, well, the model seems to be focusing on these like erroneous areas, so maybe we can do some further pre-processing on our data to help mitigate these problems. So the more like interpretable your results are, the easier it is to handle cases where your machine learning models go wrong.
I think that makes a lot of sense. But I guess what happens when the machine learning predictions seem reasonably accurate, and again, not like you said, not competing with clinicians, but how do you what are your thoughts on communicating the results of those models when you think you have a true positive, for example?
Yeah. So when you ask like communicating the results, are you speaking to like what are the metrics you want to provide, or like? Yeah,
for example, from an ECG or from a gene list, that kind of thing. I mean, what do you think is useful there for clinicians?
Yeah. So as far as like, if we look at the example of the ECGs, in terms of like the information you want to provide, then identifying like the top key important features for particular participants, that's something that could be useful for the clinicians.
Yeah, absolutely. Cool. So I guess one sort of technical question that I would kind of kick back to everybody is so when you think about this large scale analysis, Junaid, as a computer scientist, why is it useful to use cloud technology for that kind of thing instead of sort of working in an HPC environment?
Yeah, so I think the first main benefit would be like it's a lot easier when you have all of your data in one centralized location. That way, each person, they just have to bring their algorithms to that environment rather than like in an HPC where you have to allocate resources first and then everyone has to bring in their own data. So it makes collaboration a lot easier. And also it makes scaling things a lot easier as well, because you always have exactly the amount of compute resources that you need. You just spin up exactly how many instances you need. So you can use as little or as much performance as you'd like.
Very cool. Very cool. Actually, so I want to kick this back to the longitudinal data that we've been talking about earlier. Maybe actually maybe we'll segue off that point. Gemma, how do you think sort of more sort of cloud and diversified infrastructure helps longitudinal data analysis of like large public data sets or of large public data sets in this space?
So I think it's all about access, really, isn't it? I mean, something like UKB RAP, that's DNA Nexus supports, it's about providing access to extremely large data sets in an efficient way to do robust science. So whether it's longitudinal or not, yeah, that makes it more complicated, but actually the concepts are pretty similar. So I think it's about openness and access for all scientists, and so that you can facilitate that collaboration that we're all kind of voicing our thoughts on being so important. Because if you can work with people from around the globe without limitation, if you can use data from like the longitudinal analyses, the longitudinal data, if you can brainstorm, if you can create workflows and pipelines that can be shared and implemented at different times, then we're going to do some great science, we're going to do some robust science, and we're going to get hopefully towards better treatments and better outcomes for patients.
They update it too, right? I mean, you're going to have refresh rate that's faster if you have a central location for these data than if you have it distributed.
That's a really good point. So all that said, actually, I'm really tempted to take a step back. I know we've sort of gotten into some sort of thinking about some computational and technical things, but Gemma and really everybody has mentioned community a number of times. So I'd challenge you. I'd ask you sort of a softball question and then maybe a more challenging one. And the softball one being sort of what's the importance of community and working with the community in research, but particularly in cardiovascular research. But then the second question I would ask is, what do you think are extant questions out there where it would really help to have diverse members of the cardiovascular research community working on those problems together? And I'll point that to any of you.
I can kick you off if you want. So a really great example, a common disease or common cardiovascular disease, or coronary heart disease in particular, is collaboration that I've been involved with for a long time, which is cardiogram plus C4D. So when you look at single data sets alone, you really don't discover that much. You need power. So as you bring data together from across multiple partners and you grow, and you actually are kind of getting towards something usable. So all those polygenic risk scores are those tools that we use for kind of Mendelian randomization experiments and for understanding disease prediction, is because we brought people together and collaborated. And that kind of also answers your other question, which is those kind of collaborations of hundreds of people they're clinicians, they're data scientists, they're epidemiologists, they're bioinformaticians. And without that kind of differences of opinion and differences of view, you don't get such good science. You need to think about that whole package. And I think that's what kind of working together really facilitates, at least in my field. And there are other examples. I mean, working with the International Stroke Genetics Consortium, a very similar scenario. That synergy, it's just really motivating, and it's really what gets the ideas flowing. I think, you know, it's meetings like this, Ben, where you kind of start brainstorming, you start kind of thinking about things. You just don't do that if you're by yourself, or if you're in your own little world. So yeah, I think bringing people together, important topic.
I would say, I think from my perspective, coming from a pretty basic science background, I really didn't have a developed intuition for what was going to be clinically meaningful. And, you know, I think of it as there's a infinite potential realities of things that I could study in gross detail. But most of them, you know, would lack some of that aim or purpose, you know, especially when you have a context in some of these, you know, disease spaces where, you know, it's an open question. And that's sort of how I ended up getting interested in the rare variant space, recognizing what kind of a striking problem it is, and just how, with the paucity of data that's available now and probably will always be the case. And so I think sort of pointing towards the right challenges from those people who interface, you know, in a medically relevant way with the community of individuals that we're ostensibly trying to help.
Yeah, and I'd also ask the audience if there are clinicians on the call or anybody else really that has questions about some of the data science concepts we've talked about, some of the other types of concepts we've talked about. Please feel free to throw them into the chat. You know, we'd love to hear from you in the chat or the Q&A, all of that. So then let me ask a more sort of pointed question. Or maybe, yeah, let me ask a more pointed question, really, and maybe we can start with Gemma. Really, so what is the great potential? You mentioned the UKB and the UKB research analysis platform a couple of times, but what do you think the potential there is really to expand cardiovascular research?
Again, I'm going to sound repetitive, but it's about giving access and democratizing access, and kind of making that facility available so that people can engage in science where they may not have had the capacity within their home institutions to do so, and therefore some great minds are there, but actually the facilities to actually capitalize on that haven't been there. And things like the UKB RAP are providing that capacity to access the data, to use the data, and the really good initiatives for kind of early career researchers where they're kind of being given credits to facilitate them just brainstorming and using data and kind of getting their hands on data to really understand what they're talking about when they're going from concept to reality. So yeah, I think it comes down to that democratizing access. And, you know, we're all working in a big data world now. You know, everyone's an epidemiologist after COVID as well. You know, all the banks are using big data to detect fraud and whatnot. You know, big data is now part of our world, and it's part of our nomenclature, you know, on the street. So the things that are around to support that, the reproducibility, as I say, the workflows, the pipelines, the forums for people to talk about kind of reproducibility and making sure that we can do things robustly, really important. And therefore kind of, again, the UKB RAP and platforms and the cloud are part of that family of things that are necessary.
Awesome. So I'm going to take the moderator's prerogative for just a minute because I want to put a finer tip point on something that Gemma said, the first point she made, which is that if you are an early career researcher or from a developing country, there are funds available for you to do more analysis on the UKB wrap. And so really, I would advise you to think about applying for those funds and appending them to your existing research application, or if you don't have a research application yet, throw one in with the knowledge that those funds should be available for the next few years. So that's really exciting. That said, thank you, Brenton. And that said, I think we have a pretty timely question for the from the audience. So I'm gonna throw that out there. I think it sort of segues well into this discussion. And it says, from a clinician, as we develop better prediction models that start to be implemented in clinical practice, do you envision that there will be similar issues with data storage and computational power where clinical institutions will also need to think about using cloud-based services or HPC? Or is this more relevant to model development than their use?
Well, my intuition is it'd be more relevant to model development. Depends on the volume of data, I guess, that's going to be generated. And then, when you have the database, querying it, it's not the most striking challenge. And you have these central repositories for, say, variant annotation, like ClinVar and some of these other data sets that are accessible via web that don't have a huge load. You don't necessarily need to develop a neural net, on the fly to do the processing, since it's sort of pre-computed.
So Brett, but to follow that up, thinking about visualization and sort of people taking, like Junaid was talking about, taking machine learning models and making them visualizable by clinicians. I mean, it seems to me that access to the same cloud infrastructure that the data scientists are working in would be extremely valuable for clinicians.
Yeah, that's true. I mean, I still think about particular use cases in the developmental stage, but I guess, an extension is that you can maybe envision, you've developed an algorithm that recognizes some distinct features of an MRI or some X-ray. These are really information dense images, and there, whether you would want to upload it and have a sort of central processing, compute, tackle that challenge, or if you would want to do it independently. I guess my intuition is, and it's really just that I think probably early adopters are going to be developing that type of capability in a more distributed fashion, like larger hospitals and stuff being able to do that type of processing before it would be implementable in a way where it could be uploaded offsite and then processed. I would think that that's how the trajectory would develop. But that, actually something like UK Biobank might augment that strategy. I mean, it might be that there's a data set out there that sort of gets the first reasonable set of accurate predictions to come out, and then comes sort of a central repository where people want to have their patients' images uploaded and processed using the models that are developed there, rather than distributing the models out to the various hospitals or centers that are going to be generating the data. That seems to me a lower probability, but possible.
Well, so let me kick that over to Junaid and particularly Gemma, who have really sort of worked on this problem. Junaid from the sort of visualization aspect, and Gemma from sort of the organizational aspect, if you will, and thinking about deep phenotyping. And I mean, I wonder, how has, the UK Biobank model really sort of democratized science so far? But in both sort of getting to a wider swath of people, but also getting really sort of technical findings into the hands of non-technical folks.
Yeah. So first, I guess I'd want to comment on the question from the clinician. So I think the way I see it is like, with really effective clinical models, you're always in that developmental stage, because one of the most important like aspects of machine learning is continual learning, always bringing in new data and trying to update your models. And so I feel like with effective models, you're always developing, always trying to improve them. And so in those cases, I think that's where like having cloud-based service becomes really useful, because whenever you need it, you just spin it up and then make your changes, do whatever you need to do, and then deploy again. And you always want to be in like that constant cycle of improving things.
I feel like from a moderator's perspective, Junaya just hit a philosophical home run, but I'd like to ask the clinical or clinicians in the audience, if that made sense to them, and why that's important. So if you could throw that in there into the chat and continue this discussion. That said, I'd like to sort of turn the floor over to Gemma a little bit to talk about, I guess in Brett's words, to paraphrase them, really sort of UK Biobank has kind of broken the mold, right? Like of places that are collecting information and keeping them in some sort of siloed way. So Gemma, I'd ask you, like, how did that evolve within sort of UK Biobank type of stuff? And then, I know you were sort of an advisor in various capacities, but then where do you see that going in the future in terms of individual labs, individual clinics contributing data for this ongoing learning?
Sure. Firstly, I have to say I don't work for UK Biobank, but I'm one of the many scientists that work and communicate with them, because UK Biobank is really happy to hear from us. And so people that have ideas for kind of next steps and data that need to be generated, UK Biobank is also a very communicative entity, if you will. I suppose just addressing one of the points you raised as well about kind of bringing data to the masses or bringing results to the masses. And I think probably a decent example of that at the moment is the polygenic risk score environment. So a lot of work by various groups has gone into development of polygenic risk scores in cardiovascular disease, but more widely as well. And what you'll see in kind of upcoming data release from UK Biobank is that polygenic risk scores for various diseases are going to be available to use. So that puts what is something that's quite niche into the hands of people that don't necessarily understand genetics and how they were derived, but they become very usable to them. And so that to me kind of makes that transition from, do you need to be an expert in genetic epidemiology, or do I need to just understand the variable that I'm working with? And it's that kind of transition and that interplay that I think has been evolving. Now, if you take that to the extreme, which is actually in clinic, 10-minute consultation, do you need cloud computing for that? No, because that, for me at least, I would imagine that you just need a result. So for example, if you think about ECGs, just typically the algorithms that are behind them, they kind of give you a readout. There's an algorithm behind it that says this person's in atrial fibrillation. It doesn't it gives you the bottom line. And I think that kind of very usable tools need to do that. And you see that in the old risk prediction world as well, the traditional epidemiology risk prediction world, which is people want to put in their characteristics and they want to come out with a number. So there needs to be some great science and some great things that Junaid was talking about, maybe behind the scenes in the research community, but on the very, very practical level, it needs to be relatively simple to be usable, and to be able to get that public understanding of risk across as well. So I may have slightly bypassed your question, Ben. So do remind me if I have.
Actually, I wanted to follow on that. So I can imagine, a polygenic risk score. So you might send something off to be sequenced. You don't have to be a big hospital and having sequencing cores and whatnot. And you can calculate that based on the results that you found in the UK Biobank. I wonder if it might be more challenging for something that's developing a more nuanced interpretation of an MRI, where the volume of data is a bit more cumbersome. Maybe that intuition that's developed in these centralized databases then sort of, because you build a model somewhere, sort of Janaya was saying, you do build a model, you want to make that model better, but eventually you have a model that can take, these inputs. And would you think that that would again be sort of distributed like locally, like you might have it might be a feature on the software that's used to process the data already, sort of like in the case of the ECG, where you have these, readouts that are sort of processed in real time.
I think it's about who you're targeting. I think these data are really hugely valuable, and it needs people like Junaid and other people and kind of fresh brains that are looking at these data to really harvest them. But at the point of use at clinic, it needs to be relatively straightforward. So whether a specific hospital needs a cloud computing, it probably, I suspect in time, you're going to have more and more kind of mini data science centers within hospital setting. Because if you do get in, and I suppose we are heading in that direction where we sequence people, I don't know, at age 18 for want of, for various reasons about them being consented, etc. Then what do you do with that unless you can process it? And so cloud computing is going to inevitably be pivotal to that, and actually how to interpret it. So I think actually the skills development is going to have to transition as well. And I'm really impressed by the clinicians that are working kind of in the field, that actually they are definitely thinking that way. Kind of the new generation have a different way of thinking, the computational appreciation about things. Well, I'm always really impressed by the young clinicians that I'm seeing and the questions that they're asking, because I think those are the people that we really need to have on board, and they're the people that need to answer these questions. So as you say, Ben, maybe they should pipe in a little bit more in the chat.
Well, in fact, I mean, we've established that there are some on this call and they're paying attention. So I mean, we do have probably a bias sample of those really motivated clinicians who are very interested in these kinds of questions. So I think all of that makes sense. But it seems to me, and this is, I mean, I think a lot has been said here, and it's really been very motivational. But Gemma, there was a point you made earlier, which is, thinking that 2 points you made earlier that I think are kind of in juxtaposition because there's a gap. And I think that's like the extreme value of being able to communicate things like PRS, to confer risk to clinicians and others, and other types of researchers, which is great. But then also the 10-minute consultation and being able to communicate things there. And so I wonder, is one of the gaps, and Junaid has obviously done some work, and I'm eager to hear his thoughts on this too, about, pointing, not having machine learning algorithms compete with clinicians, but really sort of point them to the most relevant data. But if I can ask a more esoteric question, I mean, would integration of handheld devices, particularly cell phones, with these kinds of research clouds, in terms of thinking about risk scores, in terms of thinking about potentially relevant data points, would that really help to solve this problem?
I think we're in a world where definitely we're in charge of our own health, and we're definitely kind of moving that direction, aren't we? Where people carrying around some of their NHS records, or their medical records for the international community on their phone, and so kind of integrating apps that help monitor. And you've got your Apple Watch, you've got your heart rate being monitored. I'm not quite sure I always want to look at what's going on, but inevitably that's generating loads of data that can be integrated. As you say, there's a juxtaposition there, and I think it does come down to the fact that there's actually complicated algorithms. Okay, they might not be as complicated as kind of all the kind of ML work that Junaid and the people that he works with are looking at. But even if you look at kind of old risk scores, where you're kind of going, what's your percentage risk for having a heart attack in the next 10 years? There's lots of things under that. But what it comes down to at the point of patient contact is somebody putting in 5 numbers into their equivalent of an app and going, your 10-year risk of having a heart attack is X%. Therefore, we're going to take this treatment action. And therefore, actually, it's about decision making at that point. So, as you say, there's a gap there. How we fill it, whether it's by smartphones, whether it's by people being more informed about how to, at a patient level. I think there's, as you say, so much work to do on so many different angles. It's almost an impossible question to answer because we're all biased in our view of the world.
So, but to sum up then, I mean, is it inherently more advantageous, particularly for cardiovascular disease at this point, to be able to integrate thousands of data sets on a patient with this sort of very deep callback, with this very deep ballast? I think Brett referred to it earlier, such as UK Biobank and other large biobank studies, than to have them putting 5 inputs into a single app.
So again, you're going from a concept of population health to a single patient, and they're such different concepts. Like, you can't almost put them together. You're trying to inform what happens to a patient based on the likes of UK Biobank and large scale data, so that you have robust answers to make that insight in. And I think it's kind of that the larger data set angle, where you've kind of got to integrate lots of information and then work out what's useful at the other end. Because what I suspect that at the clinic that people don't want is conflicting information or difficulties understanding what to do, because one app says this, something else says that. Okay, what do I do with that if I'm a clinician? So people need guidance, and there has to be some sort of bottlenecking, if you like, to doing that. And it takes researchers working in all fields to make that call as what's going to be important. And there's loads of groups working on kind of making sure that what's useful, it kind of filters down to the patient level. And generating big data results and robust findings, it's going to be the best way of doing that.
So one more question, and this comes from a colleague of mine, which is, so I think the cancer community, as you mentioned earlier in the context of precision medicine, has been really informed by, one of the things that they've been informed by is this idea of molecular tumor boards, right? Where they present information, distilled information, to a bunch of oncologists, and those oncologists come back and ask for very specific stuff. Would there be a value in having some sort of a virtual cardiology assessment board, either for individuals or groups of individuals, either for the patients that they're seeing or understanding what kind of information they need, and to really bring it back to the data science community? And if people in the audience want to chip in, please feel free.
For me, for traditional atherosclerosis, for coronary artery disease, I think that we're probably not, that's probably not where we are. But when you think about things like familial hypercholesterolemia or cardiomyopathy, which is a much more rarer disease, much more focused, then there definitely is. And genomic medicine is definitely active in cardiovascular disease, just not something that maybe that I'm as familiar with. So I don't know if, Brett, you might be kind of more in that insightful in that. So I think in the common area, I can't see at the moment the value that you're suggesting, Ben, or that you're kind of questioning.
So you're saying, like, having a group of people with sort of diverse perspectives ask a series of inquiries about a particular case to try
either a particular case or
get the relevant data.
Yeah.
Yeah, I mean, I think, well, you know, I think Gemma's point about it being in sort of the rare space, you know, you see that to some extent in emails going back and forth asking. I mean, that's why, you know, me, with my background, you know, I have a PhD in chemistry and I get asked stuff, you know, I get emails, you know, a couple times a month. trying to understand these genetic variants in the context of the patient. And I think, so there's definitely already, and that is in part, especially in the rare variant space, just because there's so little data to go with. And in the more common disease space, I think part of the trouble there is, I think of it as sort of high entropy. You have so many pathways that are available that, my intuition there is that having a group of people would be helpful, but I don't think the margin on that is going to be as much, because there's so many different possibilities. As far as say etiologies, especially with things that you see in just a lot of people, where it's what's the yield of even doing genetic sequencing? Because as you guys are all well aware, you do genetic sequencing, you find things without fail. And so what you can then do with that, based on a dilution in power from over so many different disparate mechanisms. But it's just to say, I think the upside on that currently, as Jenna mentioned, is probably going to be a bit lower than in this rare variant space, where I actually think it's already it's established to some extent.
Think about how we're going to react to the data. So you can have, if you go off, do have lots of conversations. There's a reason that in cancer that's done it. There's a reason that we look at particular rare variants and whether in FH patients. It's because it affects what we do to the patient. If it's so I think kind of from a practical side, for science to be insightful, there needs to be an end game, at least for me and kind of my focus. What kind of the current era is about is almost trying to help those drug targets be developed so that we have new things in the future. And what we can really do with genetics and omics data is trying to, again, as we talked about, kind of that deep phenotyping, because there's actually particular subphenotypes that may be only understandable by using kind of multi-omics data to kind of understand that subphenotype exists. Now, it's not a good example for genomics, but it's a good example is in ischemic stroke, where you have different mechanisms. It's called ischemic stroke, but there's some really different underlying biological mechanisms. And that really affects what your trajectory might be and what the patient outcome might be, and what the risk factors and how you might want to deal with that is. So yeah, again, phenotyping really important. But I think, there's different questions at different sides of the research versus patient care angle.
No, I mean, I think the last 10 minutes have really focused around the idea that there are some cardiovascular diseases where it makes sense to communicate results to clinicians pretty much immediately and think about sort of clinician focus group, that kind of thing. And there are other things, really sort of longitudinal data challenges in common disease, where really we're focused on communicating those results to folks that are working on treatments, particularly pharmaceutical treatments, so that we can get actionable things into the pipeline.
And I think what Gemma said about it, just being maybe not right now, I think that's sort of a critical point. I mean, we're just recently being able to get large data sets to maybe begin to be powered to uncover some of these specific mechanisms, specific pathways to sort of decompose some of these more common disorders. So I would, my intuition there is somewhat bullish. I'd be somewhat optimistic about the future there.
And I don't know if you guys have kind of felt the same thing, but it's that kind of individual level data that's kind of actually bringing us forward. Because often historically we've had summary level data available, but things like the UK Biobank and being able to integrate the access to different data sets in a private way as well, so that they're not shared, but you can do the analyses kind of at the front, kind of data shields, etc. You can't do gene environment interactions without having that kind of individual level data. There's so many things you can't do without individual data, level data. And so UK Biobank and these different tools and the cloud environment really facilitate that. And I suspect that's really important for Junaid's line of work as well.
I think so, and I think I'd like to leave it there. I think that's a completely appropriate last word. Check the community at DNA Nexus for upcoming things. Also, schools, skills training, all that. Mendelian randomization just came up. There will certainly be having webinars on that at some point. Check all that out. And I'm really looking forward to the next few months. Really, we're going to be talking about a lot of very exciting use cases. So I'd like to thank the panelists very much for a discussion, and thanks to the attendees for hanging on through a long and I think very stimulating discussion. So, and thanks to Brendan for putting this together technically. There will be many more to come. Have a great rest of the week, and yeah, look forward to seeing folks for future events. Thanks, everybody.